Microbiology, Drug Inspector Exam Preparation. Prepared by Assistant Professor Akash S. Jaibaye. Topics covered. Number 1. Sterilization principles and procedures, aseptic techniques, disinfections, pyrogen and pyrogen testing. Number 2. Assay of antibiotics and vitamins. Number 3. Biochemical, serological, and virological techniques. Number 4. Biochemical oxygen demand and different media. Number 5. Microbial drugs, drug resistance, microbial limit test, and pathogenic organism of common occurrence. Section I. Sterilization, aseptic techniques, disinfection, and pyrogens. A. Sterilization, principles and procedures. Definition: process of killing or removing all forms of microorganisms, including spores. Principles. Number 1. Destruction of microorganisms by physical or chemical means. Number 2. Depends on temperature, exposure time, microbial load, and type of organism. Sterilization methods. Moist heat. Example: autoclave, 121 degrees Celsius, 15 pounds, 15 to 20 minutes. Mechanism: coagulation of proteins. Use: surgical dressings and media. Dry heat. Example: hot air oven, 160 degrees Celsius, 1 to 2 hours. Mechanism: oxidation of cell components. Use: glassware, oils, and powders. Filtration. Example: membrane filters, 0.22 micrometers. Mechanism: physical removal. Use: heat-sensitive liquids. Radiation. Example: UV for surface and gamma rays for industrial use. Mechanism: DNA damage. Use: disposable syringes. Chemical methods. Example: alcohol, phenol, and formaldehyde. Mechanism: protein denaturation and cell lysis. Use: surface disinfection. B. Aseptic techniques. Definition: procedures used to prevent contamination of sterile materials. Examples: use of laminar airflow cabinet, sterile gloves, masks, and gowns, flaming and sterilization of instruments, avoiding talking, coughing, or movement near sterile areas. Applications: in sterile manufacturing, microbiological testing, and tissue culture. C. Disinfection. Definition: destruction of pathogenic microorganisms, not necessarily spores. Types of disinfectants. Number 1. Alcohols, such as ethanol and isopropanol, which denature proteins. Number 2. Phenolics, which damage the cell membrane. Number 3. Halogens, such as chlorine and iodine, which act as oxidizing agents. Number 4. Aldehydes, such as formaldehyde and glutaraldehyde, which alkylate nucleic acids. Number 5. Quaternary ammonium compounds, which are surfactants disrupting cell walls. Factors affecting disinfection: concentration, pH, contact time, temperature, and organic matter presence. D. Pyrogens and pyrogen testing. Pyrogens: fever-producing substances, mainly bacterial endotoxins, which are LPS. Sources: gram-negative bacteria, contaminated water, and intravenous fluids. Tests. Number 1. Rabbit test. Inject test sample and observe the rise in body temperature. Number 2. LAL test, or Limulus amoebocyte lysate test. This is based on clot formation from horseshoe crab blood in the presence of endotoxin, and is the modern and sensitive test for bacterial endotoxins. Section 2. Assay of antibiotics and vitamins. A. Antibiotic assay. Purpose: to determine the potency or biological activity of antibiotic samples. Methods. Number 1. Turbidimetric or tube dilution method, which measures inhibition of bacterial growth as turbidity. For example, penicillin assay using Staphylococcus aureus. Number 2. Cylinder plate or agar diffusion method. Here, the zone of inhibition is measured around antibiotic wells. A larger zone equals a higher potency. Requirements: standard strain, suitable agar, and control standards. B. Vitamin assay. Purpose: to estimate vitamin activity using microbial growth response. Examples: for vitamin B12, the test organism is Lactobacillus leishmani, with the principle being growth promotion. For folic acid, the test organism is Lactobacillus casei, with the principle also being growth promotion. And for niacin, the test organism is Lactobacillus plantarum, with the principle being growth response. Section 3. Biochemical, serological, and virological techniques. A. Biochemical tests. These are used to identify microorganisms by enzyme or metabolic activity. The catalase test detects the enzyme catalase, such as in Staphylococci, which is positive. The oxidase test detects cytochrome oxidase, such as in Pseudomonas, which is also positive. The MVIC tests differentiate Enterobacteriaceae, covering indole, methyl red, Voges proscauer, and citrate. Finally, the coagulase test is used to identify Staphylococcus aureus. B. Serological tests. These tests detect antigen-antibody reactions. The types are agglutination, which is the clumping of an antigen-antibody complex, with an example being the Weidel test for typhoid. Precipitation, which involves a soluble antigen and antibody forming a precipitate. Complement fixation, which is used in virus detection. ELISA, which is the quantitative detection of antibodies or antigens. And finally, Western blot, used for protein identification. C. Virological techniques. These are used for the isolation and study of viruses. Examples include cell culture technique, where viruses are grown in animal or human cells. Embryonated egg inoculation, which is used for influenza virus cultivation. Plaque assay, which counts the number of viral particles. And electron microscopy, which is used to visualize viral morphology. IV, BOD, biological oxygen demand and culture media. A. Biological oxygen demand, BOD. Definition: the amount of oxygen required by microorganisms to degrade organic matter in water over 5 days at 20 degrees Celsius. Importance: it indicates the organic pollution level in pharmaceutical or industrial effluents. Low BOD equals cleaner water.
This section, titled Different Culture Media, defines a culture medium as nutrient preparations used to grow and identify microorganisms in a laboratory setting. This allows scientists to study their characteristics and requirements. The accompanying table provides key examples of microorganisms and the important drugs or antibiotics they produce. First, the mold Penicillium nototum is the source of penicillin. Second, the bacterium Streptomyces griseus produces streptomycin. Third, Streptomyces venezuelae is the source of chloramphenicol. And finally, the fungus Cephalosporium acremonium produces the group of drugs known as cephalosporins.
This slide covers 4 main topics: microbial drugs, drug resistance, microbial limit test, and pathogenic organisms. The section on microbial drugs defines them as antibiotics obtained from microorganisms. Key examples include the microorganism Penicillium notatum produces the drug penicillin. Streptomyces griseus produces streptomycin. Streptomyces venezuelae produces chloramphenicol. And Cephalosporium acremonium produces cephalosporins.
Section B covers drug resistance, which is defined as the loss of drug sensitivity by microorganisms. There are 2 main types. The first is natural or intrinsic resistance, which is an inherent feature of the organism. An example of this is mycoplasma, which is naturally resistant to drugs like penicillin because it lacks a cell wall. The second type is acquired resistance. This develops over time through genetic changes, such as a spontaneous mutation or gene transfer from other resistant bacteria. Finally, the slide outlines 3 core mechanisms of drug resistance. 1. enzymatic drug inactivation, a classic example being beta-lactamase enzymes, which break down beta-lactam antibiotics. 2. Altered drug target, such as ribosomal mutation, which prevents the drug from binding effectively to its site of action. And 3. Reduced permeability or efflux pumps, where the bacteria either decrease the drug's entry into the cell or
C. Microbial limit test. Purpose: to ensure pharmaceutical products meet microbial quality standards. Tests: 1. Total viable count, bacteria and fungi. 2. Test for specified microorganisms, for example, E. coli and Salmonella. 3. Acceptable limits: oral non-aqueous, less than or equal to 10 to the power of 3 colony-forming units per gram. Topical preparations, less than or equal to 10 to the power of 2 colony-forming units per gram. Methods: pore plate, membrane filtration, or surface spread. Reading the slide titled D. Pathogenic organisms of common occurrence. The table columns are organism, disease, source. Row 1. Organism, Staphylococcus aureus. Disease, wound infection, abscess. Source, skin, nasal cavity. Row 2. Organism, E. coli. Disease, diarrhea, UTI. Source, intestine. Row 3. Organism, Salmonella typhi. Disease, typhoid fever. Source, contaminated food or water. Row 4. Organism, Streptococcus pyogenes. Disease, pharyngitis. Source, throat. Row 5. Organism, Pseudomonas aeruginosa. Disease, burn infections. Source, hospital surfaces. Row 6. Organism, Candida albicans. Disease, oral or vaginal candidiasis. Source, normal flora. Row 7. Organism, Mycobacterium tuberculosis. Disease, tuberculosis. Source, airborne drop. Reading the slide titled Summary Table. The table columns are Topic, Key Idea. Row 1. Topic, Sterilization. Key idea, total destruction of microorganisms. Row 2. Topic, Aseptic technique. Key idea, prevent contamination. Row 3. Topic, Disinfection. Key idea, killing pathogenic microbes. Row 4. Topic, Pyrogen testing. Key idea, detect fever-producing endotoxins. Row 5. Topic, Assay. Key idea, measure potency of antibiotics or vitamins. Row 6. Topic, Biochemical or serological. Key idea, identify microorganisms. Row 7. Topic, BOD. Key idea, oxygen used by microbes, water quality. Row 8. Topic, Culture media. Key idea, support microbial growth. Row 9. Topic, Drug resistance. Key idea, microbes adapt to resist drugs. Row 10. Topic, Microbial limit test. Key idea, ensure product microbial safety. Thank you for your attention. This concludes the presentation on key topics in microbial pharmaceutical science. If you have any further questions, please feel free to ask now.